the Blueprint of Life video 11. In this video, we're going to be specifically looking at the DNA structure and function and how the work of some important scientists contributed to our understanding of the structure of DNA that we have today. So DNA stands for deoxyribonucleic acid, and each DNA molecule contains approximately 50 genes per millimeter along its length. From what we understand, DNA is a double-stranded helical molecule, and it's made of a series of subunits called nucleotides. Uh, Without the twist and the helical structure that we see, DNA would resemble a ladder, and then those sides of the ladder are made up of what we call sugar phosphate groups, and each of the steps or the rungs of the ladder are made up of bases or nucleotides, and their order determines the genes which we see on our chromosomes. So here we have some images that break down our structure of our DNA when it's not in its helical structure, which is most commonly shown in, popular media. So if we look at a nucleotide contains a sugar, a phosphate, and a base, and those bases can be either one of the four bases which DNA contains, which is adenine, guanine, thymine, and cytosine. And due to their structure and the bonds in which they possess, adenine can only join to thymine and guanine can only join to cytosine. We can see that the sugar and phosphate appear as alternating structures down the rungs of down the sides of the ladder. In terms of the four scientists that played a major role in our understanding of DNA, we have these four people here in this picture. So we have Francis Crick, James Watson, Maurice Wilkins, and Rosalind Franklin. So we have a bit of a table here that aims to describe and summarize their work, but it is part of your task to do a bit more investigation and fully understand their work. But we can see right down the bottom that in 1962, Francis Crick, James Watson, and Maurice Wilkins were Nobel Prize winners. Unfortunately for Rosalind Franklin, she'd passed away from cancer 4 years earlier, and the rules of the Nobel Prize state that the Nobel Prize can only be awarded to people who are living, which meant that Rosalind Franklin never received recognition for her work. Now, Rosalind Franklin was an expert at X-ray crystallography, and the photos that she was able to obtain of DNA showed us that DNA existed in two forms, and that one form, in fact, looked like a helix. Now, she wasn't completely convinced of this fact, and so didn't showcase her work earlier, but in fact, she was working with Maurice Wilkins, who was very well equipped at studying these X rays of DNA, and that he was, more than willing to share the work that Rosalind Franklin found about, in fact, that DNA did look like a helix. Because of this picture that was provided by Rosalind Franklin, Francis Crick and James Watson were able to publish that DNA must contain two chains of nucleotides going in opposite directions with the bases in the middle. And they did, in fact, publish this discovery in 1953 due to the work of Rosalind Franklin that was contributed by Maurice Wilkins. So here we see that for this scientific discovery to be published and understood by the scientific community, these scientists must have had to collaborate and share their information, and they did not work in isolation, which we see, especially in our work dealing with genetics. We can see already how the work of Mendel contributed to the understanding that Boveri and Sutton also provided, which all started from the work of Darwin and what he observed in finches on the Galapagos Islands. So if we just go back, this is our main structure of DNA, which we need to understand, and the sequence of our bases that follow down the middle of our ladder are in fact how we determine our genes, as it codes for certain proteins that our body can produce, which we will discuss in video 12 and beyond. So make sure you tune in for video 12. Thank you for watching video 11.